So in our DNA model, you have white, which is double ringed, which represents one of the purines, and that represents adenine. And then the light blue single ring represents thylene, which is a one ring pyrimidine, and pyrimidine, and the other two ring, which is dark blue, is guanine, and the other, and then the red is cytosine, which is another pyrimidine. And basically, you've got the green and pink bands, which represent two to three hydrogen bonds, and in our the toothpicks is our sugar, and the blue Play-Doh is our phosphates, and then the 3 to 5 structure is apparent because wherever the sugar is on the end is the 3 end, and then the 5 end is the one with phosphate.
Alright, so here we have nuclease is scanning the DNA, and it sees, oh, look at that, an obvious a mismatched pair. Oh, who would have expected such a thing? So then what it's going to do is going to tear the DNA, that part of the DNA out, because it doesn't like it, and then after it's torn it out, you're going to have DNA polymerase here, and it's going to be like, all right, I'm going to repair this section of DNA, and it's going to go by, it's going to put the nucleotides back in, except in the correct order, right? So it's going to be boom, boom, correct order. Very nice. But as you can see, there's still a gap over here. So then our old friend DNA ligase is going to come over here. It's going to come in here. It's going to snap those back together, those bad boys back together. There you have a solid piece of DNA. This is an Okazaki fragment. So here we have topoisomerase. It is preparing the DNA. It is straightening it, preparing it for helicase here, which is splitting the DNA. And after that, you have single-strand binding proteins, which will protect the nucleotides as they move out. And then you have DNA polymerase 3, and it is adding nucleotides to the DNA here as it goes along, and it's going in this direction towards the 5. Then on this side, on the lagging strand, you have primase, which is putting primers on the nucleotides to tell DNA polymerase 3 where to start adding nucleotides going in this direction. And as it does, it's going to add the nucleotides, and eventually it's going to hit another primer and stop and detach. And then later on, you'll have DNA polymerase 1. It's going to take away that primer and add another nucleotide, and then you're going to have DNA ligase seal the bond between those two nucleotides and finishing the strand on the lagging strand.